Sir, you can start, sir.
Okay, ma'am. Okay, thank you. Good evening, my dear students. Today we are going to discuss about the remaining part of the epistasis. Last class, what we have discussed already in our about. So, what is by epistasis? Otherwise called as what is by complementary gene and dominant epistasis and recessive epistasis. That things already we discussed in deep manner. Okay, and then so what is by polygenic triads? Quantitatively and also the qualitatively effect is also there. And what are the example has given? Kernel color in wheat, and this was first proposed by the name of the scientist is called as Nelson Egil. Okay, so what will be the ratio? 15:1. That things also already we discussed in the previous classes. And then the human skin colors also already we discussed. This was proved by a name of the scientist called as Davenport. Studied under the 3, what is that? Genes already we discussed that things also. Okay, what will be the phenotypic ratio here? 1:6:15 and 20:15:1 like that he obtained. Okay, how many alleles are there? 6 alleles are there. 27 distinct genotype distributions into the 7 phenotypes. That things also we already discuss, isn't it? This is the slide. Okay, how it will be obtained in the checker board, otherwise called as Punnett square. Okay, and then a monogenic and inheritance, polygenic inheritance also already we discussed. And paleotropism. What is the meaning of paleotropism? A single gene may produce more than one effect. Such type of effect is called as what is that? Paleotropism. Single gene may produce or controlled several, what is that? Effect, phenotypic effect. That type of gene is called as what is that? Paleotropism. What are the example has given in our lab? So these are the things. The particular gene responsible for the different functions. Eye, liver, and also cardiac muscles is also there. Okay, and then phenylketonuria is also there. Another one, the phenylketonuria, because it is related to the which one liver. That's why we have chosen here. So the liver is there. Okay. Second one is the Drosophila, the eye colors, pigmentations. This is also another one. Third one is starch synthesis in bee seeds. This is very, very important. There are 2 alleles responsible for this one. It also comes under the heading, which one already discussed. Okay. How it will be produced? Homozygous, how it will be produced? Heterozygous, how it will be produced? That things also already we discussed, isn't it? Okay. What are the other examples? Sickle cell anemia and cystic fibrosis, Hunter syndrome, Marfan syndrome, and Hutchinson syndrome, chorea, and lethal genes. That things also. These are the example comes under the heading. And next we will pass on to the next one is called as lethal gene. Any idea about the lethal gene, my dear students? What is by lethal? Anybody else? Ajay,
Sir, lethal means deadly, sir.
How it will be possible? How it will be possible? You are correct, but how it will be possible? Any idea?
Sir, possibly because it causes some harmful effect.
Right. When which alleles is responsible, whether dominant or recessive? How it will be happen? Whether it will be a homozygous?
Sir, recessive condition, sir.
Is it correct? Recessive condition?
Yes, sir.
Okay, very good. Exactly correct. Okay, that's why I'm asking. Good, very good. Others also, please open your mouth. Please kindly answer me. Okay. Lethal gene is one of the process genetical changes which occurs within the body. This was first introduced by the name of the scientist called as Lucien. What is that? Cunard. So these are scientist name. What happened here? All the genes present in organisms are not useful, but some of them show harmful effects. Take an example. A and B is the A, it gives a positive effect. Some of them are giving a opposite effect of the A gene. Likewise, a lethal gene is also present in it. Suppose both the gene present in the single chromosome, what will be the effect? See that all the gene present in an organism are not useful, but some of them show harmful effect and sometimes may cause death or lethality. Sometimes when it exposed, which one? Lethal gene, when it exposed, what happened? Automatically it will lead to the death condition, otherwise called as a lethal effect. Okay, this was observed by the name of the scientist Lucien Cunot. Such factor or gene that cause death or lethality in an organism are called lethal genes or lethal factors. Okay, don't forget. The lethal factors are fortunately present in recessive state and thus cause death. What is that? When it exposed the recessive manner, when it homozygous recessive character or gene present in the chromosome, it expresses once, isn't it? And end of the reaction, end of the hybridization, end of the crossing over, you will get it. When it expressed in the recessive character, what will happen? You know, that gene or that animals, that organisms completely lead to death. That condition is called as lethal effect, or what is a death. When it is expressed, you know, both the character are recessive in manner. Don't forget, my dear students, small r only, it gives the effect. Suppose if it is capital R and small r is present in it, what will happen? You know, it will not. Okay, see that. Homozygous, this is very small r is a homozygous, capital R is a homozygous, but small r is a recessive type homozygous and capital R dominant. When it both the character is both the genes are recessive manner, that time only what happen, you know, it will lead to the harmful effect or lethal effect. Okay, so don't forget it. In heterozygous condition, they may cause the structural changes. Suppose if it is heterozygous condition, capital R, small r like that, what happened? No, the structural changes will occur. A lethal allele causes death at an early stage of the development, often before birth, and so some genotype may not appear among the progeny. So that is the reason what happened, you know, during these suppose lethal alleles or lethal gene present in the parent, they may be segregate to the one generation to another generation. Suppose if it is possible, what happened now in between the F1 generation, what happened, you know, it will lead to death. So the genotypic may not appear among the progeny. Such conditions, what happened? The continuity will be suffered. Okay, see here. The lethal allele causes death frequently at an early development stage, and so one or more genotype are missing from the progeny of the cross. That is the reason. In between, there is a continuous segregation occurs. What happened in between? There is some what is a shortage or a struggle is there due to the lethal gene. What happened in the progeny? Some of the genotypic character are missing. Some of the genotype organism are missing in between the progeny of the cross. Okay. So lethal alleles modify the ratio of progeny resulting from a cross. This is also very important. Normally we will get it monohybrid ratio 1:2:1 ratio. Otherwise 3:1 ratio. Suppose dihybrid, you will get it 9:3:3:1. Otherwise 12:3:1 you will get it. Suppose the lethal is present in it, what happened? In between there is a changes also occurs. As a result, the result also suffer in the last stages. Okay, 12:1. So 3 is maybe what is that? Modified or changed during the end of the result. That has been expressed, that has been what is that? Explained here, okay, in the form of lethal gene. Okay. So some of the examples has given in the lethal genes. One is called the coat color in mice. Coat color. Already we saw, isn't it? 4 types of coat color in mice. No, in rabbit we studied, but mice only 3 colors only we were studied. Lethal factor in plant were first reported in the snapdragon is antirrhinum majus by Ebor. This is also one of the examples. Sometimes they may ask the question, lethal genes or factors were identified in the plant. This one is called as animal, isn't it? Sickle cell is also animal. Thalassemia major is also animal. Geroderma pigmentosum. Okay, geroderma. So these are all considered, it is known as animal oriented, animal related one. This one is called as, what is the antirrhinum majus is the plant. Okay, so this was just discovered by or identified by the name of the scientist called as by Ebor, B-A-U-R. Okay, you know the difference. Okay. Sickle cell anemia, what are the responsible gene? Already we discussed here. HB, yes. HB, yes. So these are the 2 things. Thalassemia major, these are the one of the disease, lethal genetic disease. Geroderma pigmentosum, these are the examples have given. Okay, we'll pass on to the next one. See here. And creeper gene in chicken. This is also another creeper gene in chicken. Hemophilia. What is meant by hemophilia? What is meaning of hemophilia? Already we studied it. Anybody else, my dear students? What is by hemophilia? Dhanalakshmi, are you here?
Yes, sir.
Okay, will you please tell me what is meant by hemophilia, my dear?
Sir, it is a recessive trait, sir.
Okay, I am expecting more information from you. What is that?
Sir, I was absent for this session, sir.
Okay, anyhow. What are the factors responsible for the blood clotting mechanism? How many factors we need? How many factors? Already we studied, isn't it? What about Ajay? Ajay, will you please?
Yes, sir. So hemophilia is also known as, so it's basically a blood clotting disease, sir, in which the blood is not clotted properly, sir.
Okay, good, super. What are the responsible factors? Do you know? What are the responsible factors? You don't know, isn't it? I will tell you. Hemophilia is also one of the recessive triads, already you know about. But what happened here, you know, there are 13 factors essential for the blood clotting mechanism. We are studying, what is that? Prothrombin, thrombin. Thrombin also enter into the next reaction. As a result, a fibrinogen converted into fibrin. Fibrin is nothing but the clotting. Only we 2-step reaction only we were studied in lower classes and as well as the higher classes also. In suppose when you are going to the deep studies, you know, there are 13 factors is there. 13 factors are particularly 3 factors responsible for the hemophilia diseases. One is called as the 8th factor, 9th factor, and 11th factor. Don't forget, my dear students, Dhanalakshmi, Ritika, and Dhanusri, and Ajay. Okay, 3 factors is the hemophilia. One is called as 8th factor, 9th factor, and 11th factor. Don't forget. Okay. Okay. So Tay-Sachs diseases, this is another one, and Hutchinson chorea is another one, and Dexter Gee in cattle, and they also this one is the, and hydrocephaly in mouse. These are the, what is the lethal genes effect. These are the examples of lethal gene facts. Okay, see that dominant lethal alleles. So just imagine. So this is the P generation, the otherwise called as parental generation. They denoted as a yellow and yellow. The genotypic ratio has given the chromosome or alleles has given here, capital Y, small y. Here also the capital Y, small y, isn't it? When it gives the corresponding gametes, what happened on capital Y separately and also small y separately. During the meiosis process, both the gametes are separated respectively. After the gamete formation, already you know about the zygote fertilization takes place. As a result, you will get the zygote, isn't it? During the zygote, what happened? You know, the capital Y forms, you know, the first one the generation will be, that organism will be lead to die. So here the capital Y is a lethal gene. Okay. So next one, so the combination will be capital Y, small y is a, it is a yellow in color similar to that of the parent. And the last one is, what is that? Small y, small y. This is called as recessive character, but it is not similar to that of the parent, but the color will be changed into non-yellow in color. It may be in some other colors. Okay. This is the dominant lethal alleles. This is the dominant lethal. Dominant means what is meant? Dominant alleles lead to the, it possesses the lethal characteristics or death characteristics. Like that you can imagine. Suppose if it is recessive means, so instead of this one, what get dead, this one is alive. Opposite manner of the dominant lethal alleles. This is recessive means same things is there, but capital Y is a live and small y is a dead organisms. Like that you have to imagine. Okay. So this is very, very important. So many times they were asked the questions regarding the frequencies, how many percentage, like that they may ask the questions. We'll practice it in calculative manner. Mathematical aspects we will discuss later. How many things is there in gene also, and pedigree analysis, that things also we will discuss later. Okay, just to note it down how it will be. For example, here the capital Y, small y is the 50-50 is there. Here also 50-50. Okay, you will divide into 4 gametes, mean 25%, 25%, 25%, 25%. During the zygote formation, what happened here? The 50%. Here the 50%. Here the, see, sorry, 25. This one is called as 50%. Okay. Likewise, they may ask the questions. Okay. I think that the part 3 will be finished. We'll pass on to the next one is part 4, principles of inheritance. Okay. Any doubt in this area, my dears? Any doubt in this part? What is my lethal gene? What is the effect of genes? How it will be expressed during the recessive stage only? Okay, but here dominant lethal gene already we discussed, isn't it? How it will be the effect in the genotype as well as the phenotype characters? What about the result in the final stage? That things also already we discussed. What are the example has given in this part? Goat color, lethal factors, and sickle cell anemia, thalassemia, major and xeroderma, pigmentosome, and then creeper gene in chicken, hemophilia, Tay-Sachs disease, Hutchinson chorea, Dexter G in cattle, and hydrocephaly in mouse. Hydrocephaly is nothing but the water storage in the brain. Okay, this is observed in the mouse. Okay, we'll pass on to the next one is part 4. Okay, so wait. Okay. So in this part, what we are going to discuss in the part 4, okay, the continuity of the principles of inheritance. Okay, what are the headings? We are going to discuss in this category, in this part, linkage and recombination. What is by recombination, my dear? What is by recombination? What is by recombination? Where it occurs? What will be the result? Any ideas, my dear? Dhanalakshmi? Recombination. No idea, isn't it?
Sir,
Oh yeah, Ritika, go ahead.
expression of non-parental gene combination in offspring.
Okay, non-parental combination in offspring. Okay, so what will be the result, whether it may be a new one or parental characters is there? The mixed together or it's a separate manner. Recombination means 2 genes are united together, isn't it? Like that. Linkage is also same, similar to that of one. Sorry. Okay. Listen here. Now we are going to discuss. Very good, Ritika. Go ahead. Linkage and recombination. It's also mostly occurs in the chromosomal aspects, whether it may be allelic, sister chromatid or non-sister chromatid, like that. Here, the collective inheritance of character is called as linkage. First of all, what is meant by linkage? Collective inheritance of character. How many characters? 10 or 100 or 1000 characters inherited from one generation to another generation. That is called as linkage between the mother to offspring, or grandparent to mother, or grandparent to grandson, daughter, like that. So the collective inheritance of character is called as linkage. The linkage first time seen by Batson and Bunnett. Note it down, my dear student. Here, the Batson Bunnett is the, what is that? First time seen by the Batson Bunnett in the plant is called as Lathyrus odoratus, and gave a coupling and repulsion phenomena. Gave a coupling and repulsion. Note it down, my dear student. Here the three question is that what is by linkage? Linkage is nothing but a collective inheritance of character is called as linkage. This is very first one. Suppose sometimes they may ask the questions. Only one type of character will be transferred or inherited to the next generation is called as linkage, whether it's true or false, like that they may ask the question. Okay. And then this was first introduced by the name of the scientist, T.H. Morgan or Sutton and Bovary, like that they may ask the questions. Don't forget linkage, Batson and Bunnett. See here, L is linkage, Lathyrus odoratus, Batson and Bunnett. Lathyrus odoratus, L is the first letter. Which plant? Sweet pea. Linkage, L. Okay. Batson and Bunnett is nothing but a square. You know about the Bunnett square already. We studied in the monohybrid, dihybrid, test cross, incomplete, dominant, yeah, whatever it may be. Okay. And not only that, and also and gave a coupling and repulsion phenomenon also given by the name of the scientist. What is that? Batson and Bunnett. Don't forget these 3 points here. 3 question is also they may ask the NEET questions. Here that there is a 3 question is there. Don't forget. But did not explain the phenomenon of linkage. But in this category, they didn't explain the phenomenon of linkage. What happened inside? Okay, this was not explained by the scientist Batson and Bunnett. The sex linkage was first discovered by Morgan. Note it down, my dear student. This is also a very important one. Last time they were asked the question. The sex linkage was first discovered by which one of the following scientists? Morgan. Sometimes they may ask the question. Sex link was first discovered by Morgan in which plant or animal, like that? Drosophila melanogaster. Coined the term linkage. He proposed the theory of linkage. Note it down, my dear students. He is not explained the Batson Bunnett. So instead of the Batson Bunnett, who stated the statement? T.H. Morgan proposed the theory of linkage. This is also another question. First discovered by Morgan, which plant or animal taken by the test sample. Third one is he proposed the theory of linkage. Theory of linkage was proposed by who? Like that they may ask the question. Which plant? It's otherwise called a sex linkage. Like that they may ask the question. Don't forget, my dear students. Linkage and independent assortment can be represented in dihybrid plant. Already we know, but this is also one of the Mendelism's law of dominant, law of independent assortment, law of segregations. Mostly, the linkage and the independent assortment can be represented in dihybrid plants only. Monohybrid, it's not possible, like that they told, isn't it? Dihybrid, don't forget, sometimes they may ask the question. Which will be represented linkage and independent assortment, dihybrid. Independent assortment, don't forget, my dear. Dihybrid plant, dihybrid grass, like that. Okay. So this is the linkage and recombinations. This is the wild type. Okay. This is the mutant types. See the diagrammatical oriented, they are having the different types. What is it? These are all are. This is the chromosome structures. Okay. The chromosomes expressed here in the round. Okay. It may be magnified in 0.0, 13, 31, 48, 54, 67, and 75, and 104, like that. Each one, it has a specific combination. The mutant type, some of the variations occurs in this category, in these portions only. So this is the wild type. this is the mutated types after the recombination of DNA from the one parent to the another parent. That is the linkage between the one chromosome to another chromosome. Okay, how the recombination is possible? That where there is a crossing over takes place, there is a recombination, there is a mutation takes place, isn't it? That is the things as given here. So in the form of what is that example? See that. Next we will pass on in the case of linkage in dihybrid, how many gametes will be provided? You know, there is a 4 gametes, 2 types of gametes will be possible, because single chromosomes, taken examples of this, this is considered as a single chromosome. It contains how many gametes? A as well as a B. Okay. And another one is a small a, small b. So here, how many gametes production is possible means capital A, capital B is one of the possible. One is possible. Another one, small a, small b is possible. 50% of AB, 50% of small a, small b is possible. Only 2 types of gametes will be possible in this type of linkage. When it take cross sections, okay, crossing over takes place from in this to here, you will get it how many? 4. But in the pure category, the same side present in the chromosome or alleles, what happened? Only the 2 possibilities given, isn't it? At the same time, see the next one. So this is the 1 chromosome. This is also another one. Here the 3rd one. This is the 4th one. Here, how many possibilities is that? In case the independent dihybrids, how many types of gametes is possible? You know, there are 4 types of gametes is also possible in this category. See the A and is a present in a single chromosomes. Another small a is also present the another chromosomes. Suppose take an example, sister chromosomes, like that you can imagine. So this is the B chromosome is there, and also another one is small b is also present the another chromosome loci respectively. So as a result, how many possibilities gametes also now? Here the 4 chromosome is there, you will get it the 4 possibilities. Capital A, capital B, here, here. And then small a, small b, small a, small b. And small a, capital B, small a, capital B is also possible. And capital A, small b is also possible. These are the 4 types of gametes also produced when, in case of independent dihybrid, you will get it 4 gametes. This is also another types. So here, A, see here. When you will get the crossing over, you will get the 4 types. So there is a combination here only. The crossing over takes place in this area only. As a result, this is similar to 25% of the parent. This is the last one is also 25% of the parent. In between these 2, you know, this is a combination of the 2 characters, the A as well as the B. Both the characters will be present inside. So you will get it the 4 times of gametes is there. This is also occur in where, you know, meiosis. Already we studied in the meiosis is also one of the reduction divisions. Already you know very about this. Crossing over takes place in the where chiasmata. Okay, the X shape, chiasma, diplotin stage. All those things already we studied in the meiosis cell division, isn't it? Likewise, recombination is also nothing but the combination of 2 genes. As a result, you will get it the mutant one. That is the things. See here. And then what are the theory of linkage? What is by linkage? Who stated the statement? How it will be produced? What are the changes which occurs in the phenotypic ratio? That things already we discussed. Theory of linkage. What is by theory of linkage? The linked genes are linearly located. First of all, you note it down. All the genes are located which manner? Linear manner. On the same chromosome. They get separated if exchange crossing over takes place between them. That is the things. That is the things already we discussed. This is the crossing over takes place in this area. So linearly arranged. Suppose they linearly arranged like this, you know, here also the linearly arranged. The crossing between these 2, you know, here the part, the particular nucleotide will also transfer to the A to B, B to A. So as a result, you will get it this type of phenotypic ratio. Okay. See that things has given here. And then strength of the linkage, note it down, my dear student. This is very, very important. The strength of the linkage infinity, 1 by distance between the genes. This is very, very important. Sometimes they may ask the question, strength of the linkage may be denoted as infinity 1 divided by distance between the genes. It means if the distance between two genes is increased, suppose there is a gene is there in a linear manner, one is present in the 0 stage, another one is present in the 15th stage, and another gene is present in the 60th, 60. So you see that first one is 15, second one is the 45 is the difference, isn't it? When the difference is distance between the two genes is increased, what happened, then the strength of linkage is reduced. Distance increases, what happened? The linkage is reduced, opposite manner. So distance decreases, what happened? The linkage is increased. That is very, very important. And it proved that the greater in the distance between the gene, the greater is the probability of their crossing over. Suppose, and it proved that greater is the distance between the genes, the greater is the probability of the crossing over. Suppose 0 to 15. Another one is also 0 to 15. The possibility is very less crossing over in 0 to 15 only. Suppose 15 to 60, there is a in between, there is a gap is there. The distance is increases, the crossing over is also increases. Know the difference between the strength and the crossing over. Distance increases, strength is of linkage is decreased. Note it down. But at the same time, the distance increases, the crossing over is also, the probability also increases. More crossing over is possible because the in between there is a 15th, 45 genes is there. They can possible so many crossing over in between the 15 to 60. But the strength is less. Okay. Do you have any doubt in this area, my dears, in this part? Because it is very important. So many times they were asked the question in this part. Ajay, do you understand?
Yes, sir.
Okay. What about the Ritika and Dhanalakshmi also?
Yes, sir.
Okay.
Yes, sir.
Okay. Okay. Any? Okay. The crossing over obviously disturb or degenerates linkage linked genes can be separated by crossing over. This is another part. Suppose it is also sometimes disturbed or degenerates. Linkage linked genes. Linkage linked means what is that? Some of the genes also united with the another one. What happened? During the separation of crossing over, what happened? It may be suffer. Something is happened. Some changes also occurs. That is the thing says given here. See that. Take an example A as well as B. Okay, take an example E. A and B, what happened? The distance will be very less in nature. So what happened? The crossing over is also very less, but the strength is also very less. But take an example of A to E. The distance is increases. The crossing over is very, what is the number of crossing over will be very high. But what happened? The strength is less. That is the things. See here, the 15% will be possible in this category. This one, 30%. Here, no linkage is possible. The distance increases. What happened? Linkage is very less. The same thing is also given here in this category, as well as the opposite manner also given in the chromosome. This is the chromosome. Linearly arranged genes also present. Okay. When the distance increases, what happens? See that the crossing over already know what are the factors affecting crossing over. If should known here, first of all, when the distances increases, what happen? The strength is decrease. But at the same time, the crossing over, what happen? Is more. Okay. Suppose the distance is decreased. What happen? Crossing over less, strength is increases. Okay. Don't forget. What are the factors affecting crossing over? This is also one of the important ones. So many times they were asked the questions. So once again and again, I will tell you. When the distance increases, already told you, crossing over also increases. The upper arrow is nothing but the increasing order. Okay. when the distance increases, see that here. Here the distance increases. Take an example A as well as E. The distance is increases. The crossing over is also possible, is also very high. Okay, that is given in the arrow mark. Distance increase, crossing over increases. Suppose when the temperature increases, the crossing over also increases. X-ray increases. The X-rays, when we are put into the X-ray process, what happened? The crossing over also increases. Sometimes they were used the crossing over process. They were used X-ray. Suppose, listen here, this is very, very important. Just like a age factor, age is increases, the crossing over decreases. Okay, this is very, very important one. Another one is that so sex, male crossing over decreases. Okay, crossing over totally absent in male Drosophila. Sometimes they may ask the question, what are the factors affecting? Likewise, that suppose the distance increases, what will be the crossing over? Temperature increases, what will be the crossing over? X-ray increases, age increases. Age is only the negative effect compared to the previous three. But at the same time, this is an example of another one, the Drosophila melanogaster. Don't forget here, the male is the example. Here, what happened, the crossing over totally absent. There is no possibilities in this category, male. What about female? Possibility is there. Maybe. Just go to the Google, search it and find out the answer kindly. Okay. Okay. Anyhow. So the arrangement of linked genes on the chromosome, how it will be arranged? Okay, already we know they linearly arranged already in our board. How it will be arranged the chromosome? There are two types of arrangement. Is there one is called a cis arrangement, another one is called a trans arrangement. Cis is nothing but both the chromosomes or alleles present the same side only. See the diagram, you can understand. Okay. Cis arrangement refers to the presence of both dominant alleles on one chromosomes, A and B, present the single chromosomes, and both the recessive alleles on the other. So already we discussed these things in the diagrammatical aspect. See here. See here. So not this one and the previous one, this one. The both the dominant alleles are present the a single chromosome. This is called as what is a cis arrangement. The recessive also another chromosomes. This is called as cis. Trans means this is called as trans. See that one A is present the one chromosome. A small a is also present the another chromosome. This condition is called as trans. Sometimes it may be two different types of alleles also present it. We will see the next diagram. I have selected there. Just to see it, you will get an idea. Okay, so see that. So this is the cis arrangement diagram. Capital A, capital B. Small a, small b. The previous slide only I have taken here because it is an example. What happened here? Both the dominant alleles or genes present the same chromosome. Recessive present the another chromosomes. What will be the gametes? Gametes already in our capital A, capital B. Small a, small b only two types. Cis arrangement. Cis. You can change it to anyone, X or Y, whatever it may be. Capital X, capital Y. Small x, small y, like that. Capital O, P, whatever it may be, whatever it may be. They may ask the question, P, Q, R, S, T, they may give so many times. Suppose if it is trans, okay, when both the recessive or the dominant allele for two triads are on the same chromosome, it is called the cis phase. However, when a recessive and dominant allele for the different triads are on the same chromosome, we call it the trans phase. Here the dominant alleles are present the same chromosome. Here dominant as well as the recessive alleles also present the same chromosome is called as trans phase. Then only we will get the four type of gametes. Okay, this is very, very important. See here, this is a trans. See that capital A is here, small a is the another one. This is the small recessive character. This is the dominant one. What about these two? Capital B and small b. This is the dominant character. This one is a recessive characters. So how many gametes is also possible here? You know, the 4 gametes is also possible here. So this is considered as a, what is that? Trans arrangement of digibrid. Okay, don't forget, my dear, this is also very, very important. So many times they were asked the question. Once again, a type of linkage. Next we will pass on to the next one is called as type of linkage. What are the 2 types of linkage? Who stated the statement? Okay, there are 2 types of linkage that have been reported by the scientist called as T.H. Morgan. One is called as complete linkage, another one is called as incomplete linkage. Already know very well. What is by complete? What is by incomplete? Incomplete dominance, already we studied. Will you please give me the examples? Ajay, do you know the answer, ma? Incomplete dominant. Dhanalakshmi, Ritika. Incomplete dominant. Yeah.
Sir, incomplete dominance, sir. Sir, the one of the example is flower, sir. Snapdragon flower.
Okay, exact one we need. It's also right. But 3 have already we discussed. Major
Sir,
one.
Mirabilis Jalapa.
Sir,
Very good.
Sir,
Very
Antirrhinium sp, sir.
okay. That one. That who stated the Mirabilis Jalaba? Dhanalakshmi or Ritika?
Sir, me, sir.
Okay.
Dhanalakshmi.
Dhanalakshmi. Good. Super.
Sir, Antirrhinium, sir. Antirrhinium
Yeah. Good.
also known as Snapdragon.
Antirrhinum majas also is there. But 4 o'clock plant, whatever it may be, T.H. Morgan, who was discovered, which one? T.H. Morgan. Say, okay, anyhow, leave it, that things. Dhanalakshmi also, good, you answer it properly. Good, well done. And also Ajay also selected 2 things is there. Gave answer, very good. Keep it up, my dear students, okay. This is the motivation for the another things also, other students also, okay. What about Ritika, please? Try to answer me. Okay. So T.H. Morgan found 2 types of linkage. They are complete linkage and incomplete linkage, depending upon the absence or presence of new combination of linked genes. Linkage is nothing but linked genes. Suppose it may be divided into 2 things. One is called as complete linkage, another one is called as incomplete linkage. Okay. Who stated the statement? T.H. Morgan. Okay, see that what happened. The complete linkage. In this category, the linkage in which genes always show parental combination. Complete linkage always show the parental combinations. It is never form a new combination. There is no, what is that? Crossing over takes place. Just imagine. See that next line is that same. The crossing over is absent in it. Same, so it may ask the question, in which linkage the crossing over is absent? Which one is shows the parental combination only, alone? Like that they may ask the question. So you have to answer it complete linkage. See that one. So the linkage in which the gene always show the parental combinations, it never form a new combinations. Crossing over is absent in it. Such genes are located very close on the chromosome. Such type of linkage very rare in nature. Example, male Drosophila, female silk moth. Okay, you must note it down. 4 point. Complete linkage. What is that? Maximum fully show the parental combination 100%. Never form new combination. There is no crossing over takes place. Okay, crossing over completely absent. But the genes are located very close on the chromosomes. This is also another one. Such type of linkage very rare in nature. Example, you note it down. A complete linkage examples: male Drosophila, female silk moth. Note it down the opposite manner. Male Drosophila, female silk moth. Okay, so note it down, my dear student. See that incomplete linkage, opposite manner it will be when a new combination also appear along with the parental combinations. So this is, what is that? Parental gene is also present. At the same time, what happened, a new combination also appears. In offspring, this type of linkage is called as incomplete linkage. The new combination formed due to the crossing over. Because incomplete is the crossing over is possible. Complete, there is no crossing over possibility. The percentage of new combination is equal to the percentage of crossing over. Note it down, my dear. It is greater than or less than 50%. Listen here. Okay. Maximum percentage is that compared to the parental combinations. This is very, very important. Percentage will be 50% more. So here you must note it down, 4 points. New combination appears with along with the parental combination. First one. The offspring, this type of linkage is called as incomplete linkage. The new combination formed due to the crossing over. So the new combination, how it will be appear? Due to the crossing over. That means the interchange between the genes from the one chromosome to the another one. Then only the crossing over is possible. Okay. It is depends upon the distance of the chromosome. Note it down there. The distance is increases, the crossing overs also increases. Okay. It may be possible 50 or 100. Okay. So the percentage is also what happened, vary. The parental combination is 50% or 25%. Parental combination means remaining 70% or 50 to more than 50% will be crossing over. New combination will be appear. Okay, that things you note it down must. Okay. See that this is very complicated, the diagram. So this is one of the example has given. I have taken. See that this is called as a complete dominant. What is the meaning of complete dominant? Means the parental character is completely expressed here only. See that? Okay. So these genes, look at the genes, then only a capital small g, small r, black body, purple eyes. Gray body, red eyes is there. The corresponding gamete is also formation takes place. As a result, he will get it the same 50% above of the parental characters only. Here there is no crossing over. There is no any new combination formation takes place. Such type of cross is called as a complete linkage. Incomplete means the crossing over also takes place. As a result, we will get it the 4 type of gametes here. See that one? Same things is there. Black body and purple eyes and gray body, red eye is there. So here the combination, what happened, see that crossing over takes place in the middle one. These two happen. What happened? The crossing over takes place within these two. What happened? The new combination also appear. Okay. As a result, see here how many things is there? 47, 3, 3. What about this one? Remaining of the previous one. Similar to the parent, but also the combination will be differ. If the crossing over also takes place, the results also different. 47, 47, 3 and 3. Okay. This is also a very, very important one. Incomplete and complete. See here. The complete linkage, incomplete. What are the difference between complete linkage and the incomplete linkage is that? The linked genes, which are closely located in the chromosome, are called as completely linked genes. Here, what happened? The linked genes which are widely located, incomplete. This is very, very important. Closely located, widely located. They do not separate and remain always together and are always inherited together only. But here, what happened? Such genes may separate at the time of crossing over. Because incomplete, crossing over is possible. Here, crossing over is not there. So, it can get inherited as it is. There is no any changes. The phenomenon of inheritance of the complete linked genes is called as complete linkage. Example, red-green color, blindness, hemophilia, myopia, etc. These are all considered the headings known as complete linkage. Incomplete linkage, see here, the phenomenon of inheritance of the incomplete linked gene is called as incomplete linkage. Example, total color blinds, retinitis, and also another one, and also last one is called nephritis, etc. These are all considered the headings known as incomplete linkage. Okay, my dear students, don't forget this category. Complete, what will be the nature? What are the examples? Incomplete, what will be the category? How it will be the arrangements, crossing over possibilities or not? What are the examples? Okay, don't forget. And then another one is linkage group is there. What are the linkage group? There are so many chromosome constitution present in a single genes. Or all the genes linked together in a single chromosome constitute a linkage group. For example, all the genes linked together, whatever in baby, skin color, eye color, hair color, and blood group, whatever it may be, they are all are present in a single chromosome. Such type is called as a linkage group. The number of linkage group in an organism is equal to its haploid number of chromosomes. Note it down, my dear students. This one. The linkage group is an organism is equal to its haploid number of chromosomes. For example, in Drosophila, the haploid number is 4. Hence, the linkage group is also 4. Corresponding numbers will be expressed. The Pisum sativum, N is 7. Hence, the linkage group is 7. Giamase, the N is 10. Hence, the linkage group is 10. But number of linkage group in prokaryote is always 1. Don't forget, my dear students. Sometimes they may ask the questions. Linkage group in prokaryote, whether 1 or more than 1, 10, 9, like that they may ask the question. Don't forget prokaryote, primitive carrion. It does not have the well-developed nucleus. That conditions what happened at the only one linkage group will be present compared to other one. Here they may ask the questions. The diamase, sometimes they ask the question 10. TP jet, whatever it may be, your aspects, you can prepare your own mnemonics aspects. Drosophila, N. Drosophila is a N. Pisum sativum, Giamase. These are all are considered the headings. Notice what is that? Pisum sativum, Giamase is the plant. Drosophila melanogaster is the animal. Prokaryote is also a, whether it may plant or animal, whatever it may be. Okay, 1. You have to split like this, then only it can be easy. Pisum sativum plant 7. Pisum sativum 7. Giamase 10. Drosophila 4. D 4. Fourth letter A, B, C, D, isn't it? The fourth letter. Just imagine like that. Your identification. Okay, for the next one. See, the same thing has given here. Okay, the Drosophila N is 4. Hence the linkage group is 4. Pisum sativum N is 7. Hence the linkage group is 7. In Giamase, the N is 10. The hence linkage group is 10. The number of linkage group in prokaryote is always 1. Don't forget, my dear. This is the important. And the name of the organism linkage group is also given. Some other examples also given in your book. Mucor 2. Drosophila 4 already we studied. Sweet pea 7. Neurospora, don't forget, Neurospora is also very, very important. One of the fungi. 7. Mice already we studied 10. This is also a important one, particularly the Neurospora and the Drosophila. So many times they were asked the question, which one is the highest one, which one is the lowest one. Likewise, you have to think. Okay, Mucor is the least one. Taken before that Mucor, prokaryote, you have to place it. Okay, prokaryote 1, Mucor 2, 3 don't know, Drosophila 4, 5 and 6 no, sweet pea 7, Neurospora 7, mice is 10. So suppose if you want more information like this, just go to the Google. Okay. Next, it's a major one, genetic map. Here we will finish it up to this level only. I give you some just an intro only, then we will pass on to the next classes. Okay. The genetic map and linkage map or chromosome map. Genetic map, otherwise called as linkage map or chromosome map. In genetic map, different genes are linearly arranged. Already we discussed each and every chromosome. The genes are arranged in linear manner according to the percentage of the crossing over between them. Suppose already we discussed the distance is increases, the crossing over is also increases. That is the things given here. Arranged according to the percentage of the crossing over infinity distance, okay, between them. Suppose they take an example, excuse me, take an example. 1 meter, 1 meter is that. Okay, 1 meter, 100 centimeter, like that, Okay, first the A gene is the last one is B, 100. In between there is a space, 1 meter is there are so many possibilities of crossing over is possible. Okay, the percentage is also very, very high. Okay, that is the things as given here. So with the help of genetic map, we can find the out of the position of the particular gene 1 chromosomes. What is the main use of this one? The genetic map can be used for the find out the position of the particular gene. For example, skin color is there. Okay, skin color, which gene is responsible for the skin color, we do not know, isn't it? Suppose we know the skin color, the present in the 50th chromosome, or 101A chromosome, or 2709 chromosomes, means we can change the color of the skin. If you find out the skin, if you have any difficulties, for example, any one of the color blinds, the fetal in the fetal stage, during the genetic disorder, suppose some of the family also having the hemophilia, color blindness, and also some other things, also sickle cell anemia, so and so. Already we discussed, isn't it? Suppose if it is find out that type of diseases, we can change it. We can rectify the diseases within the fetus. You have to collect the genes, find out the genes. 101 for color blindness, 203 for and skin color, and then another one for some other things, blood color, whatever it may, blood groups, whatever it may be. We can remove it. We can remove it with the help of the molecular scissors. Already we studied restriction endonucleases. We can substitute the positive character from the alien species. We can select the positive character. These are the harmful one, isn't it? We can remove it and then we can add the new one and replace it. What happened now? The next generation, what happened? The color blindness and also hemophilia or whatever it may be, that character did not express in the next generation. Is it possible? Yes, it can be possible. When we find out before the birth, okay, we have to identify it. So that category, what happened, the genetic map is also helpful. Not only that, and also the genetic map is also helpful in the study of the genome of the human being, not only the human being, plant, animals. There are so many type of genomes have been identified in genetic gene bank. If you want to know the any one of the organism, animals, human beings, take an example of animals like as lower animals, okay, cow or buffalo, monkey, chimpanzee. Take an example, housefly, neurospora, whatever it may be, fungi, algae, whatever it may be. Just go and check it. You will get the genetic map. Each one it has carry the. There are so many genes arranged in a manner. Have you calculated that onion contains 16 chromosomes? Okay, what are the gene is responsible? Whatever your skin color, black or white or moderate color, it is also represented in the particular genome or alleles. Okay, see that things. So it's also divided into two types, the sex linkages. Okay, we will finish it up to this level. We will see the remaining in the next class. Have to summarize the previous one. So, okay, the time is up. Okay, see here. So here we are going to discuss what is meant by linkage and recombinations. Collective inheritance of the character is called as linkage. Okay, this was first introduced by Batson and Munant Lathrace Woodwrights. Sex linkage was first discovered by the Morgan, and the plant is called Drosophila, and the coin the term is called as linkage. He proposed the theory of linkage. Note it down, my dear students. This is very, very important. Next one is linkage and independent assortment can be represented in dihybrid plant only. Note it down, my dear students. Independent assortment, linkage, dihybrid only. And these are the example, already we saw it. Okay, how it will be possible? Mutant types, how it will be possible? During the crossing over only, we can get the new combination. It is also linkage of the genome or crossing over possibility is there. So as a result, we will get the new combination, new genotype. And already you know about that the case of linkage in dihybrid. So this is also already discussed, capital A, capital B, small a, small b. That means also how many types of gametes also produced? 2 types of gametes will be produced. Second thing is independent dihybrids. This is also 4 types. Each one it has a single chromosome only. So our gene only present the single chromosome. As a result, we will get the 4 types of gametes also possible is that. That things also already we discussed. The theory of linkage, the distance increases, what happens, automatically the strength is also decreases. Distance increases, strength is decreases. Distance increases, crossing over is also increases, but strength is, what is that? Reduce. This is also very, very important. So okay, the factors already discussed. Distance increase, the crossing over increase. Temperature increase, crossing over increase. X-ray increases and crossing over also increases. Age is increases and crossing over decreases. Particularly the Drosophila melanogaster, the male, it is completely absence of the crossing over. That things also already cis and trans already we discussed. Okay, so we will see the remaining things on the next classes. Thank you for the participation. Please go through it in your book. Okay, my dear student. Thank you. Bye. Take care.
Thank you, sir.
Thank you, Ganna. Thank you.
Thank you, sir.
Thank you, ma'am. Thank you.
Thank you, sir.
Thank you, Dhanlashmi, Ritika, and so on. So, thank you. Bye. Take care.